title of my master thesis project is called The Factors Shaping the Microbiomes of Deep Sea Sponges. So the work was conducted under the supervision of Dr. Beata Slaby, the GeoMar Helmholtz Center for Ocean Research in Kiel, GeoMar. So the logo below is the one of the research cruise where I spent a month on board collecting the sponge samples. So marine sponges are the oldest extant metazoans that are great to study symbiosis in the marine environment for their tight relation with the microorganisms. So you can see a scheme of the Demosponge with a very primitive body plan where water comes through the ostium here and leaves the body through the osculum by filtering water. So at the enlargement of the internal structure, you see that the body hosts many microorganisms, here marked as in red circles. And depending on this abundance of these microorganisms, sponges can be distinguished into two groups. So the first one is the HMA, standing for the high microbial abundance, and showing the higher richness of microorganisms. And LMA group standing for low microbial abundance and presenting the lower richness of microorganisms. So the main objectives of this work are to determine the diversity and composition of the microbiota associated with sponges from Vesteris Seamount and to estimate the importance of parameters shaping microbial communities. So I'll try to answer the question to what extent these factors shape symbiont community composition. Moving to materials and methods, we have the sampling area. So that is the Vesteris Seamount located in the Greenland, central Greenland Sea. That is the largest of many volcanic edifices. So during the expedition in 2019 summer, all the samples were retrieved using the TV-guided grab and remotely operated vehicle. So here on the bathymetry map, you can see from where exactly the samples were retrieved. So the red stars corresponds to the sediment references, when the blue stars corresponds to water references, then black dots indicates the Demospongiae class, and the little sponge shape indicates the Hexactinellida class. So obtained samples were dissected and frozen until the laboratory procedures. So the next slide is DNA extraction and amplicon sequencing. And so back in the laboratory, firstly, DNA extraction was conducted on the samples, then followed the 16S RNA amplicon sequencing. And when I came to the laboratory, all the reads were already ready for me to analyze. So I used mainly QIIME2 environment to proceed all the forward reads, and then use the RStudio and Excel to do all the visualization and statistical tests. So as mentioned before, there were three factors that were implemented to the analysis. So the first one is the host identity that is represented by the lowest taxonomy level of a sponge, mostly at the genus level. The second factor is the shape. So every sponge was assigned to one of the shapes, so a ball shape, compact, cone, branching, and wave shape. And the last factor is the facies, that where Vesteris Seamount was zonated by the biological and geological differences. So we had the crust facies from the top of the seamount to 260 meters depth, then followed the shallow slope facies from 260 meters to 400 meters depth, and the deep slope facies from 400 meters to 3000 meters depth. So together with all these factors, we will try to explain the dichotomy of microbial abundance dichotomy. So some host identities were assigned to one of the group based on the previous studies, previous literature, and some I assigned myself according to the NMDS and heat map graphs using some published technique. So I'd like to start my results part with showing you the photos of the different host identities that were analyzed. So here we have the Demospongiae class, so from A to L letter, and the Hexactinellida class from M to P letter. So you can see that all the deep sea sponges vary quite a lot in size and shape, however not that much in color. And also would be interesting to show you that I and J letters, they stand for the carnivorous sponges, Asbestopluma and Cladorhiza, so that they do not filter the water, but wait and sit until the prey comes and settles on their surface. So the next slide is the alpha diversity measure of bacterial communities. So here we see that host identity and shape factors, they have the significant difference on the alpha diversity. However, the facies factor, it doesn't show any significant difference. But if you look to Giardia genus, and we see that channel index is significantly different between the crest facies and the deep slope facies. So next slide is the biodiversity measure of bacterial communities. So here we have the NMDS graph for the host identity. So we see two clusters where the samples here cluster really tightly and the other cluster is more spread and distant. And the other NMDS graphs is for the microbial abundance dichotomy. So tightly clustered samples here are the HMA. So these are actually the Craniella, Giardia, and Stellata genera. And then I move to microbial phylum level richness and abundance. So here I want to show you the heat map showing the relative abundance of every symbiont at the certain host identity. We can also see that dendrogram divides all the samples into two groups. So the first one is the HMA group, consisting of Giardia, Craniella, and Stellata, and the LMA group consisting all the rest of the host identities. So looking at this map, we can see that which symbionts are the most dominant in what host identity. However, if we look to the left-C analysis, it shows what microorganisms makes a significant difference in distributing sponges into those two groups. So we see that proteobacteria and microbiota indicates the LMA group, when the chloroflexi and acidobacteriota down there indicates the HMA group. Now I want to show you the ASV's level richness and abundance distribution. So we see that only 47 ASVs out of more than 10,000 ASVs are shared between the references and the sediments. Also interesting to show that LMA sponges share relatively higher amount of ASVs with water in comparison with the HMA group. In terms of the specific ASVs, we see that water has the lowest fraction of the specific ASVs, reaching just 50% of the total microbiome, when on the contrast, sediments present the highest fraction of the specific ASVs, almost accounting for 85%. If you look at these sponges, even though they are distributed into two groups, they present quite similar fraction of the specific ASVs, with HMA group counting 70% and LMA group showing 67% of specific ASVs. So now moving to the sum host identities, here I present you the pie charts that show the ASVs assigned to the lowest taxonomical level with a relative abundance more than 1%. So first example is the Giardia genus that belongs to the HMA group. And we can see that the most dominant ASV accounts for 3% and is assigned to the Chloroflexiphylla and the Holococoidea class and CRA202 order. And the 68% of the rest of the pie chart it presents the ASVs that are under 1% of relative abundance. So that actually indicates that the richness of the symbionts in Giardia genus is very high. The next example is Schaudinia genus that belongs to LMA group. And we see that the two most dominant ASVs account almost for 20% and are assigned to the Proteobacteria phyla and to Alpha Proteobacteria class. And this for the rest of the 43% of the total microbiome are assigned to show the ASVs under 1% of the relative abundance, and that actually indicates that the richness of symbionts of Schaudinia genus is a little lower than Giardia genus. And the last example is Carnivorous Asbestopluma genus, also assigned to the LMA group. So that we can see that the most dominant ASV is reached 24% of the total microbiome and is assigned to the Proteobacteria phyla and Gamma Proteobacteria class. And this little 8% fraction of the total microbiome that presents the ASVs assigned under 1% of the total microbiome, says that the richness of the symbionts is really low. So finally, we arrived at discussion and conclusions. So we saw that symbiont community structure clearly reflects HMA and LMA dichotomy, where HMA group shows richer and more diverse symbiont community than in LMA group. Now talking about the factors, host identities show the individual symbiont composition. In terms of shape factor, ball-shaped sponges mostly fell to HMA group, when all the other shapes such as branching, compact, cone, and waves to LMA group. So we assume that this might be caused by the difference in aquiferous system. Perhaps ball shape provides availability to form a denser mesohyl and causes slower pumping rates that favors higher microbial abundance in diversity. Where LMA sponges, they show more porous tissues and quite fast pumping rate. So the last factor, facies did not have any significant impact on the studied sponges. However, focusing only on Geodia genus, symbiont diversity showed significant difference between the crest and deep slough facies. We think that in this community composition likely correspond with the functions that are needed due to the different environmental conditions. So summing up the answering the research question, I state that the host identity had the most impact of shaping the microbiome in deep sea sponges, followed by the shape and the facies parameters. And here I want to thank everyone who listened and to acknowledge my great supervisor, Dr. Bada Slabi, for great input and support, so as giving me a chance to attend the research cruise that I enjoyed the most. Also my promoter, Dr. Professor Uta Henschel, for giving a valuable feedback and motivation, and all the marine symbiosis research group for support and big help. So, of course, all the IMBRSea family for this enjoyable 2 years experience.